Hi everyone, I'm Orlando Schwery. I'm currently a postdoc in the Weather Lab at Virginia Tech. Thanks for showing up. Where I'm going to try and convince you to use our new causal inference method to get deeper insights into your adaptive radiations. So I'm going to first real quick recap basically what I talked about last year and kind of show you where we came from, because this was a study that inspired this work. Then I'm going to quickly cover, again, like how this thing actually works, and then I'm going to try and show how we think we should be leveraging this to learn more things. So real quickly, these are the pleurodonta lizards. They're like a well-studied lizard radiation in the Americas. We had a phylogeny for those, and we had traits for reproductive mode and occurrence in the Andean Mountains. And we wanted to see whether those affect diversification, because there were hypotheses about how being viviparous in the Andes is advantageous. So it made a lot of sense. So if we use the conventional methods, the usual BISSY, MISSY, HISSY kind of things, I'm not going to show all of these, but basically they all give you a consistent answer, where they're essentially saying that both the Andes and being viviparous, and especially the combination of both of those, are really associated with the highest diversification rate. And so we think, yeah, everything is like we suspected it is. That's great. But we were kind of unconvinced by the way the data looked. And so we tried something new. And what we did was we conditioned our analyses on the observed rate shifts. So we ran analysis that found clades which are different rate regimes, and then we analyzed the effect of our traits within these rate regimes. So basically conditioning on where the shift is, how much does the trait do for this shift? And I'm kind of rush through what we found there. Essentially, there's three different things we can take out of these things. That is, in some traits, there's essentially no effect. Also, my timer's not running here, so someone yell at me when I hit the time, please. In some clades, the trait has no effect at all. In some traits, in some clades, for example, in Leolimus here, you can see that the Andes could potentially, am I pointing at the right thing? The Andes could potentially answer, like explain the whole shift, and viviparity only very little. And in some groups, we can either only explain part of the shift with the trait or nothing at all. So essentially, those are the additional takeaways we have. We know now how much our traits really help to explain the patterns we see and where the gaps are. And we would have argued that that's an improvement from the kind of insights we would have gained otherwise. If you want to learn more about that, you should totally, A, read our recent paper in Evolution Letters, and, yay, right? And you should totally go on Monday and listen to Laura Alencar's talk, who's the lead author on this paper, where she talks a bit more about the lizard side of the whole story. Now, back to us here. The next thing we're trying to bring out is basically a paper where we generalize this approach and kind of show, like, so if you're not studying pleurodonta lizards, how should you use this? And what are the different things you could or could not do with that? So we're kind of exploring the potential here a little bit. So to do that, I simulated some data, kind of inspired by the pleurodonta. Essentially what I did, I had two binary traits, X and Y. We have the X trait where we have a hidden state where one version of it increases net diversification, as you can see here, and the other does not. Then we have the trait Y where if you have it, it increases your rate. And those two traits will act additively. So if you have the X with an effect and Y together, they just kind of stack. And so that's kind of what our tree was then based on, almost 2,500 species, and we had the binary trait X observed, but we did kind of not reveal to the method that there's a hidden state in X, and we didn't reveal that Y is also there. And so I'm going to kind of assume that you know, for the sake of time, what SSE models are, that we're kind of doing trait state dependent diversification rates here, and that when you use the HSE model, you expand it to have like hidden traits nested within each of the observed trait, and that allows you to find like additional rate differences within just the trait that you observe. So if we use those on this case, what we get is, of course, that it tells us, oh, X is quite correctly actually associated with higher rates, which is true partially, but you can see the dashed lines down here are the actual rates of the different rate categories. And so you can tell that the rate we infer is kind of maybe something like a weighted average of the actual things going on. So if we use HSE, it will also correctly tell us, oh yeah, X does have an effect. Actually, yellow is higher than blue, but there's like two hidden states which kind of do different things and might have a bigger effect. But it's kind of assuming actually that the hidden state is within X. And it's not really distinguishing this from like the additional trait that we also have. So what should we do? This is where we come in. Very briefly, I've been showing you these all along. These are DAGs. You have bubbles with different factors, and when you have arrows connecting them, that means this thing affects the other thing. And apart from allowing us to kind of show what we think the causal relations are between things, it also allows us to kind of test those. And formally, you do that this way, where you like have this correlation between A and C, and if you're assuming that A affects C through B and only through B, you can test that by conditioning on B. So basically, you block any effect B has on C. And if A really only goes through B, then A should not have any effect anymore. However, if it still has some effect, then that tells us that there is another connection somewhere that we're not taking into account, and so we can come up with alternatives. Now, how do we do this in a phylogenetic context? You might be thinking of Felsenstein's scenario, right, where you have a seeming correlation between two traits over here, but then you learn, once you condition on this really weird extreme phylogeny, that the phylogeny is kind of the confounding factor that affects both of them and gives the illusion of 5? Cool. Not cool, but okay. Right, so we're asking ourselves, what are these latent clades that we should condition on? And as I pre-shadowed earlier, we just inferred rate shifts on the tree, and we use those as like what we condition on, right? So now we need to do some kind of decept test, and like, how do we condition on those shifts? Well, we make the pseudo trait. It sounds a lot crazier than it is. We basically combine the observed trait with those different rate categories to get those 8 combinations in this case. Run a regular MUSI with 8 states, which are now all observed actually on it. And then we end up again with the effect of the trait depending on which rate shift regime we're in. And we could do the thing here again where we kind of formally do the pseudo decept test to see whether something has an effect when you condition on the other thing. But it's a little bit dry, and we're out of time, so I knew that to begin with because it would take quite a while to dig through all of that. So we can also look at it a bit more intuitively and thinking of it as some kind of sister clade analysis. So the question is, does the trait have an effect? It does, but only in some of the clades and not in the others. So we could consider that a hidden state, right? Because the X's that don't have an effect are a different hidden state than the X's that do have an effect. And now the question is, are there still unexplained shifts? Well, there are, right? Because we have this here and this here, where X has an effect but not fully, and X has nothing here, but there's still a shift. So we might want to consider whether that is another trait. It could be yet another state within X, but if you kind of look at it, it's very tempting to think, oh wait, this is almost the same effect size in both of those. so it should be the same thing. And if this is already another hidden state in X, they are mutually exclusive, right? So we might update our hypothesis accordingly. And we're not done at this point, right? We need to investigate what really is there, what makes sense. But we now know the distribution of the unknown thing that we're looking for, because we know where the rate shifts are and we know where the traits are within those. And so we can kind of focus ourselves on like, well, is the next trait I'm wanting to look into distributed that way? If it's not, then maybe it's not a good candidate to look at. So yeah, going forward, again, use DAGs because it's really useful, condition on the patterns to the causal inference, as I just showed. But also going forward, now that you know the effect of one trait, when you look at the next trait, you could try and condition on the effects you already know. And even before you condition, you can just compare them and you can see like, oh wait, is this new trait explaining a shift that the other one didn't? Oh, it kind of is, right? And it inversely, like, the old trait explained a shift that this one can't explain. There may be still a bit of a gap, so you might have to look into that. But we would look at that and be like, oh, this could be a very promising scenario, and we should like focus on that going forward. I'd thank the Ueda lab and the Muniz lab and a bunch of other people who were kind enough to discuss this with me. Again, if you haven't read our paper yet, check it out. Talk to me about other ideas I have about this. Find me online. And I'm just going to leave with this slide because the Ueda lab is here with a bunch of other exciting things. So you should put them on your schedule if you haven't yet. Thanks.
All right. Hello, my name is Andrea Halling, and I just finished my PhD at the University of Colorado and started a postdoc at the University of Utah a couple weeks ago. I just finished like my 8th cup of coffee, so I'm gonna try and like talk slow, but my brain's like, bleh. So sorry in advance for that. And today I'm gonna talk about isotopes. The title of my talk, like on the schedule, is like, does multicellularity have an isotopic signature? And it turns out if you have a question as your title, the answer is always no. So I changed it. So now we're just going to look at the isotopic perspective of division of labor. So throughout my PhD, I was really interested in this transition that life went from being single-celled to being multicellular, and specifically becoming really complex, like how we are today. And when it comes to multicellularity, there are a lot of multicellular forms that we see today. So here is a really cool picture of nobrion, which is a stromatopile. It's a mixotroph, so it can do photosynthesis, but it can also like phagocytize bacteria, which is cool. We got some Spirogyra. It wouldn't be an evolution conference if I didn't put a picture of some yeast up there. And then this is like a snowflake, well, like a choanoflagellate colony. So these are all really cool examples, but something that makes complex multicellular really unique is that there is this aspect of division of labor. You have multiple cell types. So humans have like over 200 types of cells, and that's really fascinating when it comes to an evolutionary perspective. So I'm just really interested in how that happened and how can you detect that from a macroevolutionary perspective. How can you actually find when this evolution happened? And so in my PhD, I came across this paper that I was like, this is really interesting, this idea that division of labor might happen through plasticity. If you have a clump of cells and they have the ability to do different things on their own, then maybe if they're exposed to different types of environments when they're in a clump, they'll start to differentiate. And so with that kind of idea, I started thinking about this in terms of algae and the algae I work with. And Chlamydomonas, a single-celled algae, can do phototrophy, it can do photosynthesis, but it can also utilize acetate. So if you start to have like clumps of cells, so assume each of these big circles is a clump, then you start to have these really interesting options for how those clumps of cells might utilize their resources. So maybe all of them can use CO2 and they're all doing photosynthesis. Maybe some are doing photosynthesis and they're producing some sort of sugar that the others can eat. Or maybe they're just using different carbon sources from the beginning. So again, with this in mind, I was wondering how I could apply this into like a testable system. So here's a beautiful phylogeny from Lindsay et al. Looking at the Volvocene algae. And this is a very cool system to look at because they're all fairly closely related, but you have a range of complexity, essentially. You have Chlamydomonas here that's single-celled. Then you start to have groups of cells, but they're all the same cell type. And then you get into like spheres and differentiated cell types. And this makes for some cool things to look into. So I started off kind of choosing some bookend organisms. And the reason, so if you go back here, this is Chlamydomonas, this one is Volvox. And Volvox is very cool because it has these germ cells that do reproduction, and then it has these like terminal somatic cells. And hypothetically, they're doing different types of metabolism. So we can sort of probe at this by using carbon isotopes. So the question for this part of what I wanted to look into was, does multicellularity and specifically division of labor produce this unique isotopic signature? And the prediction here would be that if metabolic division of labor is present in Volvox, because maybe some cell types do more heterotrophy than other, we would expect less fractionation in Volvox than clammy. So let's pause and take a step back and talk about some of these words. For those of you not familiar with isotopes, here's your carbon isotopes 101. There are two main stable isotopes of carbon. Carbon-12 makes up the vast majority of all carbon, and then you have carbon-13 that just makes up 1.1%. When it comes to photosynthesis, photosynthetic organisms preferentially use the light carbon. And this is really awesome because that can be detected. So if this box is a heterotrophic organism, it is what it eats. So if you have some sort of, sugar here, and this sugar is made up of some ratio of carbon-12 and carbon-13, what you actually see in the biomass of that organism is going to be exactly what it eats. If you have something that does photosynthesis, then the CO2 that this organism is essentially eating, it's going to preferentially select for the lighter carbon. And this makes the pool of available CO2 different than what you see in the biomass. So you end up getting lighter biomass. So again, this preference can be detected by comparing the ratio of the biomass to the ratio of the environment, its source, essentially, and that's called fractionation. So again, back to the question, breaking this down, what this means is what we'd expect to see if you have division of labor of metabolism is that Volvox, if it's doing less overall photosynthesis, it's going to fractionate carbon less. It'll be more similar to its available carbon. Whereas if you have Chlamydomonas that's doing more photosynthesis proportional to Volvox, it will be, there'll be more fractionation. Okay, so what does this all mean and how did I test this? I pretended I was an isotope geochemist for a little bit of my PhD, and this was actually really fun to kind of figure out a system to look into this. So what you can do is you can take a culture and hook it up to an instrument called a Picaro that will continuously measure CO2 concentration and fractionation in the headspace of the culture. So as it's growing throughout the entire growth curve, you can get this continuous measurement. And so on a chart here, on the x-axis, we have residual fraction of CO2. That is looking at like you have the initial amount of CO2 in the headspace. As it grows, it's going to be sucking down CO2. And so over here at 1, that's 100% of the CO2 is available. And as you move through it, it's eating it as it goes all the way down to 0. So that's what the x-axis is. On the y-axis, you see this delta 13C. And this is now looking at these ratios. So this is going to be the ratio of C13 to C12 in that culture headspace compared to some known standard. All right, so once you inoculate the culture, you start here, the blue is the substrate, that's the CO2 in the headspace, and the green line is what's being incorporated into the biomass. So as it's using up CO2, there's going to be this curve. And if you linearize this, essentially, that the slope is what we're calling epsilon. This is going to be our fractionation. So to simplify this, the steeper the slope, the more autotrophy or photosynthesis is doing, and the less steep, the more heterotrophy. So if it's not fractionating at all, then you're not going to have any sort of slope. So we set this up, and this was a really fun thing to kind of play around with and set up. If any of you have done isotopes, you know that it can be a lot of work to process a sample and run it through like an elemental analyzer, and you get, like 15 data points, and if it makes a line, you're like, this is great. We ended up getting a ton of data. So this is for chlamydomonas, and each little black dot is a value. So you get these beautiful lines, and if you run it in triplicate, you get these beautiful parallel lines, which was really exciting. And you end up getting this epsilon value of about 15.8 per mil. And per mil is just the notation of fractionation. And then when you look at Volvox, I don't know why it's black like that, you get a little bit different value. So again, when you put them on top of each other, you start to see that there is this difference in slope, which is very cool because that means that there's more fractionation here in chlamydomonas, meaning that relative to this entire population, it's doing more photosynthesis on average than Volvox, which is really cool. If you come back to the kind of prediction, this ended up being what I thought, which is very cool. So conclusion, the next step is this is a potential perspective to kind of study division of labor, like metabolic division of labor. And I'm working right now, I've done it on a handful of other of the algae in that phylogeny, and I think it'd be very cool to look at it phylogenetically and see how that looks. But yeah, that's kind of what I'm working on, and using this method of looking into metabolisms based on morphology throughout my postdoc. So with that, shout out to my lab mates. I don't know if Dinah Davison is here, but she is the one that sent me the Volvox, so I'm very grateful for that. And with that, I will take questions if anyone has them. Yeah.
Really cool. I'm curious, if the heterotrophic taxa are consuming sugars, wouldn't those sugars likely be produced by a phototrophic taxon?
You mean in nature or in these experiments?
Yeah. So in nature, if you're looking at the actual fossil, wouldn't you expect fractionation to decrease as you go up trophic level?
Oh, that's an interesting question. Yeah, I'd have to think about that. The other huge caveat here is that fractionation can change a ton depending on metabolism. So it's cool that this works out this way, but there's definitely a lot more to look at. If you have layers of things that eat things and produce things, how is that impacted? But yeah, cool question. Awesome. Okay, thank you.
Hi, everybody. My name is Sean McHugh. I am a PhD student in Michael Landis's lab, interested in phylogenetic comparative methods, methods development, niche evolution, and historical biogeography. I'm here to present to y'all a novel method that I've been developing to both model the realized and fundamental niche envelopes for a single climatic variable in continuous niche space, both anagenetically and cladogenetically. But first, here's some cool animals. Here's a monk parakeet, and here's a cane toad. And they're both really good examples of animals that can really thrive well outside their native range, right? So with monk parakeets, it's in Staten Island, and cane toads, well, you can pick a place pretty much. But they were both extremely successful and good indicators, both species that are probably succeeding far outside of their realized niche into other parts of their fundamental niche. And what do I mean by that? Well, I'll go through a little small cartoon here of a cartoon salamander. And this salamander, this is its fundamental niche, the range of abiotic conditions that it can thrive and persist in this case in terms of temperature and precipitation. In this case, like this salamander and like many other species, this salamander is only really found in a small subset of that, that larger yellow envelope that's in blue, and that's designated as the realized niche. Now, the fundamental is truncated to the realized niche by a whole host of factors, in this case, in the upper precipitation quadrant from dispersal barriers, say like a mountain range, and in the lower portion from a competing species, biotic interactions. These are two examples of a lot of different factors that can come into play here. But it's really important to consider this, especially given we usually only have realized niche data, and we consider our phylogenetic comparative methods in terms of the realized niche, occurrence data and where species occur. However, what we're really interested in is both when species are moving and evolving through niche space. And so in this case, I jumped a little ahead here, but in this case, we're showing with the same color coordination as well in a single univariate climatic niche envelope, yellow being fundamental, blue being realized. We're seeing here an instance where both are moving. This make-believe species here is shifting upwards in temperature both in terms of its physiological tolerances and where it's actually occurring. So it's both moving and evolving to access this portion of niche space. But a lot of times, we just have species that are simply moving. The niche is already sufficiently wide, and it's just other barriers got released, and it's able to move within its niche space. And again, with realized niche data, this is really hard to get at. And our fundamental niche data is far from great. We all know this. But can we actually start maybe getting at this a little? And there have been a few recent studies and more data coming to light that I think makes it so we shouldn't just leave it on the cutting room floor. Take, for instance, with, with a lot of plants found in many arboretum and botanical gardens well outside of their native range. Those occurrence days have been shown to very recently by Laughlin and McGill that many species have potential niches well outside of their native range, and that often these are overlap niches far more conserved than is expected. And then also, we have a lot of good thermophysiology data, especially when utilized with mechanistic niche modeling such as Niche Mapper, other tools incorporating natural history and microclimate and everything else that we know about these systems. We're even often able to project out and realize species are often underfilling their climatic niches, especially in terms of their warmer niche breadths, as was shown by Nikki Moore and et al. In this Nature Eco Evo paper, and showing a cool example of Leolamus, one of my favorite groups, and a good amount of data with them. Now, is this data sufficient, or is this great data? Nah, no way. I mean, it's still pretty limited. Maybe you only have one limit, or just the other, the upper breadth or the lower bound. But it's still data that we shouldn't ignore, and maybe it can help us see in some instances maybe something's not evolving. Instead, it might be just moving across niche space. But we also run into another issue. Oftentimes, our phylogenetic methods, we're really limited to only like single niche envelopes. And that's still like great work and really cool stuff, really important methods that we still have a lot to learn from. But in this case, I mean like an envelope being like we have like maybe a position in niche space and then a breadth around it. And then I'm showing here this little example with a parent lineage. You can have cladogenetic events. That's been some recent really cool work done. Theo Gabro and others. And then we have an example of like two daughter lineages also stochastically evolving from that point. But what is the envelope? What envelope are we looking at here? Is this the realized niche? Is this the fundamental niche? How do we discern when things are moving versus evolving? Even with what little data we have, how can we reconcile the two? In this case, this is where I guess I come in with a novel method that I've been trying to implement, where we look at fundamental and realized niche evolution, both anagenetically and cladogenetically, in a Bayesian model that I've implemented in Julia thus far. Called RFC, Realized and Fundamental Continuous Niche Evolution. So let's look through this thus far. With this model, building off of what I was showing before, we have, I'm adding an extra layer. This here, say this is the realized niche envelope. And building off of it, we add the fundamental niche. Both are evolving continuously through niche space at the same time, with the realized niche always being constrained within the fundamental niche along this given niche axis. Allowing for cladogenetic evolution to also occur either over the realized niche, the fundamental niche, both, or neither, in a continuous range of scenarios. So first, let's start with what are the characters here? How do I fit all this together? Not easily, and probably not the best way, but it's start. So for fundamental niche, this is actually in climatic niche space, such that we have a niche breadth, which can range from almost 0 to any arbitrary value, a position within this continuous niche space. And then for realized niche, we model it as a relative to the fundamental niche in order to make it so we can model all these things together reasonably. So with the realized niche, we have a proportion. What is the proportion of the fundamental niche? We logistically transform this so we can model this in continuous niche space in Brownian motion as with the other characters. And then the offset, which is how skewed or offset is the position of the realized niche relative to the fundamental niche, either from I had an error, was 1 and 1, hopefully it's -1 and 1. So 1 is the most extreme to the upper end, and -1 is most extreme to the lower end. And for a few examples here, here's just a few different examples of niche envelope combinations that we can get here, where proportion being 1 and offset being 0, well, they're pretty much identical. You have instances where the proportion is 0.3, offset is 0. It's smaller and just centered in the fundamental niche. Offset of -1, it's fully to the maximum, the bound of the fundamental and the realized are up against each other. And +1, the bound of the upper of the realized and fundamental are right up against each other. And so we model all these using multivariate Brownian motion in a continuous model. And this is the androgenetic evolution. This allows us to get at a few different scenarios that would be kind of hard to tease apart otherwise. So we can look at scenarios where either the realized or fundamental niche is evolving quickly or slowly, such in this case where it's both slowly, so the realized distribution over this climatic gradient is very conserved, and the fundamental niche gradient is conserved as well. You could have instances where maybe your realized position relative to the fundamental niche is relatively conserved, but your physiological tolerances are changing. Maybe you're jumping between multiple different microhabitats, having a lot of different habitat use strategies that are changing rapidly, but within your macroclimatic range, it's staying relatively constant. You could have instances where a fundamental niche is really conserved, but your realized niche is shifting rapidly within that, where you're moving much more, you're evolving, and the niche is very conserved. Or you could have some crazy thing where both are going very fast in some complex history. But these are things that we can actually start evaluating the probability of, or rather really characterizing the uncertainty around. That's more the name of the game with this, I'd say, at this point. And also for cladogenetic work, I really want to build upon what other folks have been doing. There's been a lot of work on this very recently that is really cool. Theo Gabbro, I mentioned before, his asymmetric Brownian motion model, he's right there, thanks man. It's really cool work. And for my realized niche evolution model, it's building heavily off of that in its parameterization. Go check out that poster Monday. And likewise, we're estimating these cladogenetic events, seeing different event types over the tree using a data augmentation approach from Sarah Swiston, who's doing similar work with continuous range evolution. Go see their talk. Yeah, whatever that is. I can't see from here, sorry. But it's going to be a great one, I promise. But anyways, with that, let's look at some of the cladogenetic events we can see here. So we can have anything from serious subset splitting here, where you can have splitting either over the realized niche, the fundamental niche, where the other one is remaining constant, or splitting across both. This can line up with many different scenarios we could imagine, either where it's like geographic speciation along a climatic gradient, ecological speciation where both tolerances split, or somewhere in between. And these are just the most extremes. You can have a continuum of both to some degree. This isn't like the only 4 discrete scenarios. This is a continuum amongst these 4. Likewise with subset symmetry, where you could have one daughter lineage inheriting a subset of the other lineage. And that could subset either in terms of the realized niche, the fundamental niche, or both. Okay, I realize I'm messing up on time here probably. So just quickly on like a small example, I wouldn't say I'm really learning anything super new about this group. I just wanted to show this as an example of the types of questions you can ask and the types of things you can test with this type of model. And the example being North American pinus trees. I really did a small subset of the tree here, and the occurrence data of realized niche is informed by occurrence data that we have that from GBIF and other sources, and fundamental niche data being, or potential niche data being from Arboretum data from the BGCI. And again, this is far from actually getting the full fundamental niche. This is just expanding out to a larger potential niche. But even with this, where we're having a larger envelope than the realized niche, the big question to me is, when can we discern when things are actually evolving, that yellow line is fully moving, versus how often the realized niche is just moving within that? And we fit this, and it actually mixed pretty well. We got our fuzzy caterpillars, if anyone's into MCMCs. I don't know, thank God. But anyways, one thing that we got that was interesting is I just took our ancestral state reconstructions very quickly, and this is like a pretty hacky way, but just to get a sense of it, looked at the relative displacement of the realized niche versus fundamental niche over both optimum and position. I transformed those offsets and proportions back into positions and breadths. I think of optimum position, same thing, you get what I mean. And we just looked, when are we seeing where the realized niche is being displaced far more from the start of a branch to the end of the branch relative to the fundamental niche in terms of both position and optimum. And we're seeing a lot of instances, it's about the same. It's about the same. The green means is both are being displaced similarly. We're seeing instances, though, where the realized niche is being displaced far more than the fundamental niche through time, where I'd say, some evidence that there's a bit more movement going on than evolution, perhaps, or something where it's something warranting further study, for sure. Likewise, we're seeing some instances here where the fundamental niche position or breadth is changing much more rapidly or being displaced far more than the realized niche. Granted, I take that less seriously because in this case, the realized can bounce up and down within the fundamental. So it might not be there might be more displacement or more actual evolution in the realized niche than we're seeing here, but we're adding in correlation, ways of measuring correlations between these characters and better ways at getting at this part. But this is just the first pass to see when are things maybe moving more than they're actually evolving. And then last part here, cladogenic events. So I took our continuous parameterizations at each of the nodes, and I kind of binned it into key categories here, whether we're having splitting versus subsetting of daughter lineages in terms of the realized niche, fundamental niche, or both. Lighter colors are splitting, I believe, and darker colors are subsetting. And over the board, it's pretty messy, and we're not expecting to get perfect estimates for this for every node. That's a bit crazy, but we would like to characterize the uncertainty around this and maybe find a few nodes where some signal is clear. And now's the time for everyone's favorite game of cherry picking. And I'm going to pick a node here that I like, that we see a clear pattern here of subset symmetry in this one node, where the daughter lineage going to Tida, and I'm not being able to read the other one there, but you get the one that branches in the next two taxa, that we're seeing a considerable subset in terms of both the realized and the fundamental niche. Both are truncating down in that instance. And that's something that you wouldn't even be able to get it characterizing before these tools. So, okay, I have this crazy model. We have to do a whole lot more simulations before we get anywhere close to really throwing this at a lot of data. But there's a lot more I want to see in terms of coverage, validation, seeing how much fundamental niche data is sufficient to be able to actually get estimates before everything blows up into an uncertain mess. But also, more deals of incorporating tip error, other ways of incorporating uncertainty around those where we won't get that fundamental niche data, not just treating it as, oh, those bounds are the same, how much are they actually sampling random error and characterizing the uncertainty around those points more so than just saying, oh, the fundamental and realized niche are the same there. Or even better, try to start inferring the fundamental niche at those tips to some degree and characterizing uncertainty that way as well. That'd be the name of the game, using those joint inference approaches such as NEMO and B-Fine shameless plug, and other approaches that allow us to jointly infer ecological niche models and phylogenetic models of niche evolution jointly. And with that, huge thanks to everyone in the Landis Lab. It's been a pleasure. They helped me a lot, and thanks to y'all for putting up with this. Much appreciated. I'll take any questions. Thank you.